right.
Okay, let's talk about synthetic genetic circuits. Synthetic genetic circuits are artificial systems made of DNA that are designed to control and regulate the behavior of living cells. They work by taking advantage of the cell's natural gene expression control machinery to turn genes on and off in response to specific stimuli. Now, the arsenic biosensor I showed you is a simple synthetic genetic circuit. It runs a simple program which determines if arsenic, then express green fluorescent protein. This type of synthetic genetic circuit is called a buffer gate, and its activity can be described by the truth table shown at the bottom of the slide. Truth tables are ways of demonstrating or showing the activity of a synthetic genetic circuit that does logic computation. I'm going to walk you through this truth table because we're going to show a bunch of these in this presentation, and I want you to be comfortable looking at truth tables and familiar with what it is they're trying to tell you. So this truth table again describes the activity of a buffer gate. In the case of the arsenic biosensor, the input is arsenic, and an input value of 0 denotes no arsenic present in the water that you're trying to measure arsenic concentration in. And an input value of 1 is a toxic level of arsenic in the water. Now, the output is expression of green fluorescent protein. So when there's no arsenic, you should also have no green fluorescent protein. But when there is arsenic present in the water, you should produce green fluorescent protein. So your output value should be 1 when your input value is 1. Okay, so that's a truth table for a type of synthetic genetic circuit called a buffer gate. And it, again, is a really simple type of synthetic genetic circuit, but it's powerful, right? It's enabling you to produce a bacteria that responds to a contaminant in the environment and produces a visible signal. Now, what if you want to make a slightly more complicated biosensor that responds to arsenic and lead in the water? Let's say you want it to produce green fluorescent protein in response to either of those two contaminants. Well, you could use a slightly more complicated synthetic genetic circuit to achieve this goal. The type of synthetic genetic circuit I would recommend using for this is called a logical OR gate. OR gates, again, respond to two inputs. And in this case, your inputs could be arsenic and lead. So A might be arsenic, B might be lead. Your output would still be expression of GFP. So you should get GFP when either arsenic or lead or both are present in the water. So by just increasing the complexity of your synthetic genetic circuit a little bit, you now get a bacterium with a broader set of capability. Now, let's say instead of just detecting arsenic or lead in the water, you actually want to remediate or remove these contaminants from the water. You could potentially express a set of enzymes that produce a chelating molecule or something that removes the arsenic, let's say, from the water. An example of a molecule that can chelate arsenic is 2,3-dimeraptopropanol. This compound has a high amount of sulfur in it, and so you maybe don't want to be creating it all the time. And you might want to only be producing this compound when there's arsenic in the water and when there's a sufficient amount of sulfur in the water, because otherwise you may be making enzyme that's not able to produce the chelating molecule. So you should, in this case, probably use a logical AND gate, where your two inputs are arsenic and sulfur. Again, the sulfur is there so that you can produce enough of the dimeraptopropanol in order to chelate enough arsenic to be meaningful. And you should be expressing the enzymes that make that chelating compound only when there's arsenic and sulfur in the water. Okay, so by using a logical AND gate, you can get a sort of smart remediator, a smart cell that is able to start to remediate arsenic or work on remediating arsenic only when it's there and when there are enough resources there for the bacteria to do the remediation that you want it to do. Now, I just showed you a buffer gate, an OR gate, and an AND gate, but there are actually even more types of logic gates that you can build into living organisms in order to control their behavior. There are 16 different 2-input logic gates that allow you to take 2 different inputs and determine what to do based on the presence of those 2 inputs. And you can build even higher order logic gates too, where you have let's say 3 inputs and you only produce your output gene when all 3 inputs are present in the environment. And so these types of logic gates are really powerful. They're only one type of synthetic genetic circuit too. I believe Professor Endy showed you some oscillators. That's a different type of dynamic synthetic genetic circuit. But what's true for all of these different synthetic genetic circuits is that there are many different ways to build them, and there are lots of biomolecules that you can take advantage of in order to construct a synthetic genetic circuit. So we're going to talk on the next couple of slides about building logical AND gates. I'm going to show you designs for 4 different AND gates built with different types of biomolecules, so you can get a sense for how bioengineers are thinking about creating synthetic genetic circuits inside of living cells. Okay, so this first AND gate is built with transcription factor and chaperone pairs. So the design of this AND gate uses 2 different inducible promoters. One promoter is turned on by the presence of arabinose, that's a type of sugar, in the cell, and the other promoter is turned on by a type of antibiotic called tetracycline. And so when the arabinose promoter is activated, it turns on expression of a protein called SIC-A. SIC-A is a transcription factor that would bind a third promoter called p-SIC-A and initiate expression of the gene downstream of SIC-A. But SIC-A, when it's produced, is not folded correctly. For whatever reason, the bacteria is not able to fold it correctly. And so remember we talked about how protein structure is really important for function. Well, when it's not folded correctly, it can't bind the DNA and it can't initiate expression of the output gene, which is RFP, a red fluorescent protein. SIC-A needs a chaperone called INVF in order to fold correctly. So the second input, the tetracycline, drives expression or initiates expression of INVF. This chaperone protein then helps SIC-A fold correctly, and it's only when you get both SIC-A and INVF expressed in the cell that you get a functional transcription factor that can then turn on this SIC-A promoter and initiate expression of your output gene RFP. So that's the design of this AND gate. Its activity was assayed in E. coli. And so in this graph, you can see increasing amounts of tetracycline along the bottom and then increasing amounts of arabinose up the side. And you can get strong expression of RFP, your output gene, only when there is both a high concentration of arabinose and a high concentration of tetracycline in the E. coli cell. And so this is exciting. That means that type of AND gate built on transcription factor chaperone pairs is functional. It's not the only way to build an AND gate. You can actually take one transcription factor and split it in 2 and drive expression of 1/2 of the transcription factor with one input promoter and the other half with another input promoter. So only when you then get expression in promoter A and promoter B that you get both halves of the transcription factor being made. Once both halves are made, they can bind together and activate an output promoter And in this case, it's driving expression of a different fluorescent protein called cyan fluorescent protein. These are just different colors of fluorescent protein. The activity of this type of AND gate was assayed in a type of mammalian cell line called U2OS cells. So I think it's both exciting that we're able to build AND gates with a different type of biomolecule, in this case, a split transcription factor, and in a different type of cell, a mammalian cell, which is very different from an E. coli bacterium. Okay, so you can also build AND gates with a special type of protein called a recombinase. Recombinases are proteins that invert segments of DNA. So the first recombinase in this AND gate design is BXB1. BXB1 expression is stimulated by a molecule called acyl homoserine lactone. So this is just a molecule you can add to the cell. When you add it to the cell, you get expression of BXB1. BXB1 then looks for its recognition sequences, which are segments of DNA in this cartoon indicated by little red arrows. When it finds the DNA that it recognizes, it will actually invert the part in the middle. And so the DNA in between these two recognition sequences is a terminator. The terminator will stop progression of RNA polymerase. So this promoter should be attracting RNA polymerase and then initiating transcription of an RNA. However, when the polymerase hits the terminator, it'll fall off and it won't be able to express the gene that's downstream, which is GFP here. So BXB1 will invert this terminator sequence and essentially allow the RNA polymerase to progress to GFP, or at least it would progress to GFP, except that this design has two terminators in it. The second terminator is flanked by recognition sequences for a different recombinase called PhyC31. PhyC31 expression is initiated by a antibiotic, and this is the same one as before, tetracycline. When PhyC31 is expressed, it will invert the second terminator so that when you get expression of BXB1, which flips the first terminator, and PhyC31, which flips the second terminator, you can then get an RNA polymerase that is able to express or read through this segment of DNA and express your downstream gene GFP. So it's another way of building an AND gate inside of a living cell. This type of AND gate was tested in E. coli and found to function very well. The last type of AND gate that I'm going to show you is built with proteins called proteases. These proteases cleave proteins, and so there's no transcriptional control in this case. All three of the other circuits that I showed you, all three of the other AND gates, were all dependent on changes in transcription in order to get changes in expression or GFP signal. In this case, we're going to have control at the level of protein stability. Okay, so a protease, again, cuts proteins at very specific sequences, very specific amino acid sequences. And the protein in this case is going to have a GFP fused to a degradation tag and a TEV protease cut site, and then another protease cut site for HCV protease and another degradation tag. Now, these two degradation tags will cause GFP, the protein, to be degraded really quickly. So you never see fluorescence signal in a cell, even though it's making GFP protein, because the protein is so quickly chewed up. Now, the TEV protease is going to be expressed in response to your first input, and it will cut the protein just upstream of the GFP part so that it removes the degradation tag. It will essentially cut off this degradation tag. Your second input is going to be this HCV protease, and it will cut off the other degradation tag. So when you have both TEV protease and HCV protease expressed in your cell, you'll cut off both degradation tags and get a GFP signal. So in this case, you're going to be getting AND gate-like logic just by cutting a protein in specific locations. So this is just showing you fluorescence intensity in HEC293 cells. That's a type of human embryonic kidney cell line. So these are human cells. You can see strong GFP signal only when both your TEV protease and your HCV protease are expressed in the HEC cells. And so this type of AND gate is just another example of a logic gate or a genetic circuit being built with a biomolecule. What is true for all of these is actually that you can build diverse genetic circuits with the same tools. So the same kinds of protease tools can be used to build synthetic genetic circuits that perform other types of logic. This is a not AND gate, so it has actually exactly the opposite type of behavior as an AND gate, where it's on in all cases except where both TEV protease and HCV protease are expressed. There are, implies, not implies, and exclusive OR gates, just all different types of logic gates that you can build using the same type of protease tools and using the same type of recombinase tools. You can build diverse types of logic gates as well. And so these different biomolecules can be used to build different types of synthetic genetic circuits. And through all of this, you may be wondering, what's a concrete example of where I would use a logic gate? Like, why would I go through the trouble of building these things? Well, one example is here. A synthetic genetic circuit can be used to increase cancer treatment specificity. So in this case, we're talking about a type of cancer treatment called a CAR T. So these are T cells, which are immune cells that are activated to basically kill cancer cells within your body. We've engineered the T cells to recognize the cancer cells as foreign and then start breaking them down. And the T cell needs to be very specific and only start killing cancerous cells, because if it starts killing other cells within your body, it can cause a lot of side effects and be potentially detrimental to your health. And so an AND gate can be implemented in order to help the T cell very specifically kill cancer cells by very specifically recognizing things that are only on the surface of cancer cells. And so by having 3 different input signals, the T cell is able to more specifically kill cancerous cell lines. And this graph on the right is just showing you that it's able to do this very specific targeted killing of cancer cell lines when it has a 3-input AND gate, or when the 3-input AND gate is used in T cells. We're going to talk more about the mechanism of T cell-based or engineered T cell-based cancer therapies in our Cancer Week. But I just want to show you that this type of 3-input AND gate can be useful for refining treatments, which is, I think, an exciting application. Okay, now tuning gene expression, changing how much transcription or translation is going on within a cell, can change the behavior of a synthetic genetic circuit. So just want to show you that with a really simple synthetic genetic circuit called a NOT gate. In this case, it is just flipping the buffer gate around. So if you have an inducible promoter, which we're denoting with this gray arrow here, that's turned on by something like tetracycline or arsenic, any of the inducible signals we showed before, instead of expressing a GFP signal, it will express a repressor. This is a type of transcription factor that then turns off a promoter. And so if you turn off a promoter that's upstream of your output gene, in this case, a yellow fluorescent protein, then you'll get a transfer function or a response function that looks like this, where as you increase the amount of your input, let's say you increase the amount of arsenic present, then you will decrease the amount of observed fluorescent protein, because as you increase the amount of input, you make more repressor. That repressor turns off your output. So you get something that looks like this. Now, what we care about in these types of synthetic genetic circuits are the dynamic range. What is the difference between the on state and the off state? You want a big dynamic range, or a big difference between on and off for most biosensors or most synthetic genetic circuits, because if you produce, for example, just a little bit of fluorescent signal in response to arsenic, then it can be really difficult to see is my bacteria fluorescing or is it not? Am I making, is there arsenic in the water? Is there not? Whereas if you get a really big signal, if you get a big dynamic range, a big difference between on and off states, then it's much more obvious that the bacteria has detected something in the environment that it's trying to tell you about. And then the other thing we care about is just the threshold, right? This is the point at which a genetic circuit switches from being on to being off or being off to being on. This you can think of as the threshold at which there's the bacteria recognizes that there's arsenic in the water and starts responding to it. You can imagine that at a really low concentration of arsenic, maybe the bacteria can't even detect it. It can't even tell. And so that threshold is just where the switch point is from being on to being off. Now, if you change different parts of this NOT gate here, then the circuit will behave differently. So for example, if you change the strength of this second promoter, this red output promoter, then you can change the kind of whole circuit's output. So if this black line is the original circuit, and you increase expression or the activity of this red promoter, then you can shift the entire curve up. You basically will be producing more output protein all the time. Even as you try to turn it off, you'll still get more than you would have originally. And if you decrease this promoter strength, then you can shift it down. The other things you can do are change the degradation rate of the fluorescent protein, let's say in this case. If you start increasing the rate at which this protein is degraded, let's say you put one of those degradation tags on, but not a very good one, just one that's kind of okay, you can reduce the amount of fluorescent protein that's being, that's stably produced in the cell at any time and shift this curve down. By changing the ribosome binding site, you can actually shift the curve left and right and change the thresholds. So basically what I want you to take away from this is that there are many ways that you can tune synthetic genetic circuits in order to tune the activity of the, or the behavior of your living cell, your biosensor, your remediator, whatever it is that you're trying to build, your CAR-T therapy. These curves, these are not experiments. These are actually mathematical models. So we actually can predict what would be the impact of doing something like changing promoter strength. We understand enough about how biological systems are functioning in order to do this type of prediction. And so if you are a bioengineer and you're designing a synthetic genetic circuit, you might build a mathematical model first and then say, okay, what parameters do I need in order to make the circuit function correctly? How strong of a promoter do I need? How strong of a ribosome binding site? Stuff like that can help guide the construction of synthetic genetic circuits that have different performances in living cells. Okay, the other thing I'll say is that it's not just the expression control elements that matter, but actually the part choice affects circuit performance. So I showed you a couple of different types of AND gates, one of which was built with transcription factors and chaperones. And in this case, the AND gate that gets built is very sensitive to changes in both the chaperone and activator expression levels. And so what do I mean by that? Well, it responds dynamically to changes in these things. If you have input 1 and input 2 pulsed into your system over time. So let's say you have a bacteria and it's living for 3 hours, and then you add inducers, both the tetracycline and the arabinose. Those were the two triggers for promoter 1 and promoter 2 to initiate or become active. So you add both of them together, then you will get expression of both your chaperone and your activator at the same time, your transcription factor. Those things will bind to the promoter and initiate expression of your output gene. And so you'll see an increase in the amount of your output over time, and then it will hit eventually a saturation. And when your chaperone and your activator, when both things are still being expressed, it will stay on. But then if you wash away the arabinose and your tetracycline, these two input molecules, eventually you'll stop making the chaperone and activator, and then expression of your output gene will turn back off. So you get this kind of dynamic response to the inputs. If you have them temporally separated, then you'll actually never see the input or the AND gate turn on, right? If first you make some activator because you have one of your inputs present, then you stop, adding that molecule, and later you add the other molecule, you will get, chaperone being expressed, and then later on in the cell, you'll get the activator being expressed, but they were never expressed together. They were never expressed at the same time. And so they won't be able to activate expression of your output gene. And so this type of AND gate shows you when both inputs are present at the same time, when the bacteria is experiencing both of them together. But this recombinase-based AND gate does something different. It makes a permanent change in the DNA. Remember, one of the recombinases will invert a terminator, and the other invertase will invert the other terminator. But these inversions, these are permanent. So they are permanently changing the DNA. So if you express, and they take a long time to initiate relative to the expression of your activator and chaperone. So if you have both promoter 1 and promoter 2 triggered at the same time, if you start expressing both recombinases at the same time, you will get expression of your output gene slowly turning on as these recombinases catalyze this inversion. But what's really interesting is that if these things are temporally separated, you will get the first invertase expressed early on. It will invert its piece of DNA, its terminator that it recognizes. And then, that invertase, the promoter expressing it may shut back off. You may stop making this recombinase, but it will have already done its inversion thing. So now this piece of DNA is sitting here with one terminator inverted and the other one still preventing expression of GFP. But if you put the other input into the cell, then you can get inversion of the second invertase and expression of your output gene. So this AND gate is actually reporting whether or not your two inputs were ever delivered into the cell, not whether they were delivered together. So it's just an interesting nuance of how when your choice in what biomolecules to use to construct synthetic genetic circuits affect the cell's behavior. And so there's this big race within synthetic biology or bioengineering to figure out what are all the different types of molecules I can use to build synthetic genetic circuits. And when I build circuits with these molecules, how do they behave? So I just want to leave you with that thought. And then I have some reflective questions. So I want you to think about what are other biomolecules that you could use to build AND gates? Maybe these are biomolecules you've heard of in your biology courses, or maybe you're just, dreaming. Maybe you're thinking if only there were a biomolecule that did this thing, we could build a really cool AND gate with unique dynamic responses or, other unique properties. I want you to reflect for a little while on what would be an interesting biomolecule to build a logical AND gate with. I want to think about why tuning gene expression affects circuit behavior, and then what situations you think synthetic genetic circuits could be helpful in.
All right.
We're going to explore the idea.
Okay. Well, that was great. I think I need to probably re-watch a lot of this kind of stuff to actually get so familiar and quick with this kind of stuff. All right, got a lecture down.